So it's a pleasure to be here, even though I have to follow this very tough act. But I've been at Michigan for a couple of years now, and I think I've been really looking for the opportunity to get out here to sort of meet some other people that are essentially doing complementary things. And in particular, Oski's been a good contact to have. And I also mentioned this in the context of Michigan has recently started a regenerative medicine collaborative, where we're really trying to facilitate teamwork, team-based science in areas related to regenerative medicine. And so I think my talk today is really going to center around some of the technologies that we've been using for cell-based therapies. We're going to use islet transplantation as a model because that's one of the systems that we've been using quite regularly, but hopefully you can see that if you're not doing diabetes work, hopefully that can be a model system for you to think about the cell types that you'd be interested in working with. And if you are interested in diabetes, well, that's great, let's talk. So the whole idea, I think, for regenerative medicine is to have a progenitor cell and try to coax that cell down the fate that you would like. And so you can do this by controlling cell interactions, materials for both cell adhesion, mechanical properties, drug deliveries, and nanoparticles. And so really it's the whole idea that I often refer to what my lab does as systems tissue engineering, where there isn't one magical factor that's going to get this whole system to work. You need to create the system itself to essentially provide the right combination of factors. And then also I think we've recently started to try to engage systems biology to try to figure out what are the right factors and then sort of combine those opportunities. So as I mentioned, I wanted to start with diabetes. I think particular attraction of it is one of the more advanced cell-based therapies that are out there. It's still experimental in nature, but it actually is being done clinically. It's just that there's not a large supply of islets, but it is done. I believe the last I had checked, there are roughly 9 approved cell-based therapies at the FDA. So this is not necessarily one of those because the cell source and other things are not quite worked out, but definitely it's been used clinically. The Edmund protocol was very key in terms of really getting this into people to recognize the opportunity. In that trial, it was shown here that you essentially would take a pancreas from a donor, isolate islets, and then the recipient would have the islets infused through the portal vein where they get trapped within the sinusoids of the liver. And the particular novel part of the Edmund trial was the immunosuppressive regimens they would use that actually allowed the patients to become insulin independent by receiving 2 to 4 donor transplants. And at the same time, ultimately, these patients remain insulin independent for a period of time. And while this plot may be somewhat discouraging, that it's down to essentially 10% by 5 years, more recently the immunosuppressive regimens have been tweaked such that now they're around 40% engraftment. So it is a process that's moving forward, although I think our argument is that it's still a challenging process. And in part, I think, as the engineer in me would say that the liver is not a natural home for an islet. The islet is normally sitting within the pancreas. It's normally very highly vascularized. The islets occupy about 1% of the mass of the pancreas, but get about 10 to 50% of the blood supply. And so they're very highly vascularized, which sort of makes sense of putting them in the vasculature. However, you can imagine it's coming in from the portal system, and so therefore the oxygen concentration here is relatively low. The ECM that they're seeing is very different. There is an inflammatory response, Kupffer cells and others that essentially develop that often lead to a steatosis that is part of that inflammatory response. And all in all, that's, again, not the right home. And not only is it not the right home, there's very little you can do to change this because the liver is the liver, and the transplant surgeons I talk to say, no, you cannot change the liver, despite my suggestions to the contrary. So we've been working with Bill Lowe, who is an endocrinologist, Shrimrung Lowe, who is works in transplant immunology, but she's a nephrologist professionally, and Steve Miller is an immunologist who's working in autoimmune disease. And we've been working on this concept of actually transplanting islets, what we refer to as these microporous scaffolds. The islets themselves are typically in the range of 100 to 150 microns. There are some lower, there are some larger, but generally they're in that range. And we end up making a scaffold that's, you can see here, that has pores that are in the range of 250 to 425 microns. And they're made out of the biodegradable, same material that biodegradable sutures are made out of, the polylactide coglycolide. And so we're able to essentially simply load the islets onto the scaffold simply by just seeding the solution, and the islets fall within the pores of the scaffold and become trapped. And so why this is essentially useful, or one of the steps in the process, is that you're actually going to be spreading the islets across a space. So rather than having them clump and aggregate and then be competing with each other for nutrients, you're now actually distributing them across the surface, and we can transplant this into the peritoneal fat, is where we've been doing. So the omentum is simply our target site for this. And the material actually adheres to the fat relatively well, and you can just wrap it up as you're wrapping up a burrito. So it's a very simple process, doesn't require any sutures. And so in addition to spreading things out, the adhesion to the peritoneal fat provides essentially direct apposition to the tissue, so you actually get blood vessels growing in quite readily. And the material is also degradable, so it's actually going to go away with time. So it's really going to be a deposition of natural tissue around this, and the material goes away. So I think that's sort of the general feature of it. I do want to take 1 second just to distinguish this from much of the work that you hear in the field. When we started working with these microporous scaffolds, people would say, oh, people are doing this left and right. But really, this idea of having a porous scaffold that allows an integration with the host tissue is very different than what most of the field has done. Most of the field has been using encapsulating biomaterials that actually prevent the integration. They keep the immune system out, which obviously, as Ariel indicated, you want to do that in some instances. However, our premise for this is that, again, the islets are 1% of the mass of the pancreas, get 15% of the blood supply. These need to be able to sense blood glucose and distribute insulin very readily. And by isolating them from the host vasculature, you're actually depriving them the ability to really sense and respond, and also changing that microenvironment very dramatically for them. And so that's why we have chosen not to do an encapsulating system. So I'm going to sort of walk you through this sequentially, because I think our approach has really been, let's tackle one problem at a time. And so our initial work was done with syngeneic transplants. So we just wanted to say, we want to take the immune system out for now. We'll deal with it later on, but let's see if we can just get engraftment to happen. So we would take islets from a donor, seed them onto the scaffold, transplant them into peritoneal fat of a mouse that's been treated with streptozotocin to make it diabetic, and then we can monitor over time. And you can see here the example of the islets that are seeded onto the scaffolds and trapped within the pores. First aspect you note here is, I'll use this bottom right one as an, maybe I can point to these over here. So the blue is indicating of insulin. Within panel E, you can see that in F, the dotted line outlines the blue, which indicates PCAM, and then in G, you can see that there's a tomato lectin stain, which actually came from infusing the tomato lectin into the vasculature. So these islets are directly hooked up to the vasculature by transplanting them onto the scaffold, which allows them to, again, sense that blood glucose and then distribute their insulin as needed. But let's look at this at a longer time. I mentioned we really kind of want to create this environment. One of the key features for us has been that when you isolate islets from the pancreas, you use a whole bunch of proteases that actually degrades the basement membrane around the islet, which also can be something that influences its viability and its function. So we did something very simple. We coated this material with Collagen IV or laminin, or in this case fibronectin, to actually try to reconstitute that basement membrane towards what those cells might want to see. So just taking you through this, you can see time across the x-axis, blood glucose, and the y-axis. The dashed line is the upper level of what's considered normal blood glucose levels. When you transplant islets, if you don't transplant islets, you can see pink, that the mice become diabetic and essentially stay that way, and then we actually had to euthanize those animals. However, when you transplant islets on these scaffolds with these different coatings, you can see that the blood glucose eventually comes down below the dashed line. It's sustained that way out to about 300 days, at which point my MD PhD student said, I really want to graduate. And so we let him euthanize the animals, but not before he actually explanted the peritoneal fat where he transplanted them. And you can see the animals became diabetic when you remove that tissue. So the other aspect I'll point out here in this arrow you can see between 100 and 150 is that's really where the scaffold falls apart. After that point, we cannot see any signs of the scaffold, and it's degraded. Before that point, you section it, you can see the material. So these cells continue to function throughout the whole period of time that the scaffold is degrading, and also continues to function after that scaffold is gone, because it's created a host tissue around it that supports its function. And then the last part I'll mention in the slide is really more easily seen here, is the extracellular matrix protein that you use matters tremendously. You can see here in Collagen IV coating, all the animals became non-diabetic within 4 days, whereas it took more than 50 days for the fibronectin and laminin-coated animals. And if we use the fetal bovine serum, which obviously was just our control, you can see that actually not all the animals actually converted. So that environment is really critical. Follow-up studies had really pointed to, by using Collagen IV, we really enhance the viability of the cells, their metabolic activity, and their glucose-stimulated insulin secretion. So all these things are enhanced by having the right extracellular matrix components there. That really led to what we believe was a better survival of the islets upon transplantation, and that better survival led to essentially a better function in the post-transplant period. So in addition to doing mouse islets, we've also used immunocompetent mouse model and actually transplanted human islets on these scaffolds. You can see them as actually on the surface of the scaffold where they just get trapped within the outer 500 microns or so. And essentially here are the applications comparing kidney capsule, which serves as the gold standard for mouse. And you can see here that the transplant of the microporous scaffolds mimics exactly what we see with the kidney capsule. And so really why this is really impactful is really this idea that the kidney capsule may be the gold standard, but it's really not a translatable site. And so we wanted to get the islets out of the liver and get it to an extrahepatic site such that we could actually begin to think about controlling this environment and being a more robust system for engraftment and for delivery. Now this was done, as you can see here, with 2,000 islets, which is consistent with what you would normally see in the kidney capsule. But we did want to push this a little bit further. And we pushed this by saying, could we actually deliver factors from the scaffold itself to try to change that environment? Obviously, we mentioned Collagen IV as being a factor, but there's also Exendin 4, which is something that's actually known to stimulate glucose-stimulated insulin secretion with islets. And we're actually redesigned the scaffold so that we kept the outer region, which was that microporous region where we could actually seed islets onto it, but we actually developed an inner disc layer that we could actually load the Exendin 4. We could actually get a sustained release for about 30 days from that inner layer. And you can see here, by using this scaffold system with a Exendin 4 delivery, we actually reduced the number of islets transplanted by about 25%, down to about 1,500 islets that were transplanted. And you can see here, without the Exendin 4, these islets are stressed and ultimately do not really continue to function for long term. However, by doing this sustained delivery Exendin 4, we're able to get a better engraftment and better function for longer periods of time. So I don't know that these would continue to function long term, but it does show the opportunity of actually delivering the factors of the short term within that environment. So the reason that I particularly mention this is, while it may not be ideal for mature islets, we're also beginning to translate this work to actually using pluripotent stem cells, where you actually want to differentiate them down to a beta cell lineage. And so there's been publications that have actually taken pluripotent cells and taken them to pancreatic progenitor cells, or even farther. This is stage 4. There's stage 6 and 7, where it's actually more immature beta cells. But we actually have been different. We do the in vitro culture to get to the cells to stage 4, and then we transplant these cells on the scaffold. You can see here they're loaded with these pancreatic progenitor cells. We transplant them, and again, most of this work with transplanting these pancreatic progenitors has been done in the kidney capsule. And you can see here the kidney capsule essentially is the middle curve here. And when you transplant them in the kidney capsule, they actually do have a relatively robust function and engraftment, but that kidney capsule is not a translatable site. And so if we go from the kidney capsule and just use a PLG scaffold in the peritoneal fat, the C-peptide levels are relatively modest. However, if we actually use an Exendin 4 and we transplant the peritoneal fat, you can see significantly elevated C-peptide levels by these cells. We actually be able to both stimulate the cells and actually try to drive their development down the beta cell lineage by delivery from the scaffold. And you can see here the human C-peptide is increased with the Exendin 4 delivery, and we actually see more monohormonal cells with Exendin 4 delivery as well. So it's really taking us in this direction where we're actually working with these progenitor cells, manipulating the environment, trying to push the differentiation of the cells. And for those of you that may not be working in the islet field, I did put in a single slide here from a paper that we had from last September, where we're working with Jason Spence, and he's been using with human lung organoids. And he'd been transplanting human lung organoids just subcutaneously or in the kidney capsule. And when he used Matrigel as a delivery vehicle, he would consistently see, the tissue begins to grow, but you would get a very immature development of the cells. However, when we actually transplanted his human lung organoids on the scaffolds that we have, we actually saw a much more organized structure, and actually we're getting more mature airway structures to develop that you can see here, where you actually have the basal cells are actually present in the proper context and organization that seems to mimic a lot more of what you would actually see in a human lung. So while I don't have any more slides on this, I think this is just the opportunity of actually taking progenitor cells, trying to push them and support their differentiation and support their development in ways that you can't really do by just transplanting cells alone. So I'm going to transition to the other part that obviously if you're thinking about both diabetes therapies as well as going beyond the animal models, and particularly I think this is the opportunity to talk about doing these in large animal models, the idea of trying to protect the cells from immune destruction. And that's, I think, there are mouse models where that is readily done. I am not aware of widely used large animal models where you actually have them immunosuppressed and can do similar types of studies. So this is going to be something that needs to be done. The work that we've been doing with the islet transplant, we are now actually taking into non-human primates with Jose Oberholzer at UAC, and we're just applying immunosuppression regimens that he's using clinically. But really, obviously, we'd like to get away with that. That's been a major focus of JDRF, who we've been interacting with on this. And so, as you can imagine, the immune system has two obviously key components. There's the innate response and the adaptive response. I think our work with the syngeneic cells suggests that the innate response isn't a limiting factor in terms of transplantation. What we see in terms of how the innate cells respond to our scaffolds, the islets still are able to engraft and function. So in a syngeneic setting, they're not so difficult as to compromise islet function. However, when you start to go into an allogeneic setting, you can imagine these cells are going to get more activated and are actually then going to be presenting antigens such that you would actually be activating the adaptive response as well. That's in an allogeneic setting. And then also if you're talking about an autoimmune disease such as type 1 diabetes, there's actually going to be pre-existing T cells that are going to be essentially responsive to the cells that you're transplanting as well. So that's obviously no small challenge in terms of what we're doing. So we're looking to say, how can we begin to address this? So I've been working with Haval Sherwan, who's at the University of Louisville, with actually taking advantage of Fasl. So Fasl is, many of you may know, is referred to as a death ligand, and it binds the Fas receptor, which is referred to as a death receptor. And so on activated T cells that express the Fas receptor, when they encounter the Fasl ligand, it normally induces apoptosis in those T cells. And so Haval, for many years, has been working with this Fasl ligand variant that he's developed, where it's a streptavidin to Fasl ligand chimeric protein. And he actually would go through and take cells and decorate them with biotin, and then he would incubate them with the streptavidin Fasl ligand to actually take the cells and basically be able to decorate islets with Fasl ligand. And then he would then transplant them into the kidney capsule. And so what we've done here is actually just take his very similar strategy of actually taking the Fasl ligand modified islets, and we put them onto the porous scaffolds. And hopefully you can see here across the bottom that the by transplanting the islets that are modified with Fasl ligand into the peritoneal fat. And I will add, I forgot to mention that this Fasl ligand modification of the cells is accompanied with a 14-day rapamycin treatment, after which case it's discontinued. But in this case, if you take the islets with the Fasl ligand and the short course of rapamycin, you can see that these islets survive and function for the better part of 200 days without needing any sustained immunosuppression after those 14 days. And if you don't have the Fasl ligand, you just have rapa. After the rapa is discontinued, you see that the islets reject pretty shortly thereafter. And so in this case, both the allogeneic and syngeneic cells actually behave very similar to each other. And because the islets are integrated with the host tissue, the glucose tolerance test that's very commonly performed, you can see that the native islets as well as the transplanted islets really are able to restore blood glucose levels on a very similar timescale. So what I think is very intriguing to us in terms of this strategy is this local presentation of Fasl is able to create sort of a local immunoprivileged site. Now, what we wanted to do is try to avoid this idea of all the cell manipulation that happens. And so we really began with a strategy of, can we take the scaffold and can we modify the scaffold with Fasl and then just take islets and transplant it on the scaffold. And so essentially we started out by biotinylating the polymer, making it into microspheres, and then using these microspheres to then fabricate a scaffold. And then we can see the islets on the scaffold that's now modified with Fasl. The Fasl presented on the scaffold in this way is still able to induce apoptosis in cells that actually express the Fas receptor. And you can see here, when we transplant islets on these scaffolds, we see that approximately 90% of the transplanted islets are able to function long-term when given that short course of rapamycin, whereas without the Fasl, they actually reject relatively early. Interestingly, this middle curve here, we actually decided to give a very high dose of Fasl and try to cut out their rapamycin, and we had approximately a third of the islets actually engraft long-term without any immunosuppression whatsoever. So we're continuing to pursue what we could do to sort of really get rid of this immunosuppression, but this idea of having a local effect in terms of trying to manipulate immunological responses within and near the graft seems to be encouraging in terms of what's possible. Now, along the way, I was also able to work with Shren Rong and Steve Miller, whose picture I had shown earlier, in terms of really trying to say, can we do something about the autoimmune response as well as the allogeneic response, but targeting it not from just creating a local site, but actually having more of a systemic effect. And I've often thought of this technology that Steve Miller's been working with since the mid-70s as really sort of retraining the immune system not to respond to specific antigens. And so therefore, you're not just creating a local site, you're sort of systemically doing away with the response to specific antigens directly. And so Steve his first paper was in 1979. We're using cells as the carrier for antigens, and then a mere 30 years later, I come along and we say, let's replace the cells with particles as the carriers for those antigens. And ultimately, rather than putting the protein and peptides on the outside, we've actually been encapsulating the proteins on the inside of the particles. So let me just give you a brief sense of the efficacy of this, and it's really, to me, a really interesting story. So this is essentially working with essentially an autoimmune model. And the way that we did this autoimmune model is we use these BDC 2.5 mice, which have a T-cell receptor specific for chromogranin A. And so we isolate the T cells from the donor mouse, from the spleen. We take those T cells and we activate them in vitro. And then we do an adoptive transfer into the recipient. And you can see here, if you adoptively transfer these activated T cells, that within a couple days of the adoptive transfer, you see the mice become very diabetic very quickly. And essentially they're going in and essentially destroying the islets within the pancreas of the recipient. So to counter this, these really activated cells, for which we're delivering about 2 million cells into each animal, is we actually, chromogranin A essentially is this peptide P31. So if we use a P31 and we take that peptide and we put it onto or into these nanoparticles that we're making, and we give a single injection of the particles shortly after we deliver, adoptively transfer the cells, you can see the animals actually do not develop diabetes. However, if we use the wrong peptide, you see that the animals actually do develop diabetes. So we're not knocking the immune system down for everything as you would with an immunosuppressive drug. What we're knocking down is the response to very specific antigens that are associated with the rejection of essentially the islets in this case. So I thought this was remarkable that this could happen so quickly in being able to reduce that response. And really, I'd say that we did one additional part where we actually depleted regulatory T cells from the graft, and when we deplete the regulatory T cells, you can see that the grafts actually reject, the mice develop diabetes after the adoptive transfer of the cells. So these Tregs being present are key to actually the development of the sustained resistance or sustained tolerance in the system. So to just give you, in the short amount of time that I've had, I know I've glossed over a lot of details, so just to give you a sense of how we think this is working, there's been specific immunotherapy which has been delivering peptides or proteins into the blood for actually almost 100 years now, and you're able to actually tolerize to antigens in that way. The challenge you can imagine is that if you have a tolerized animal, you also have the potential to do an anaphylactic reaction that will lead to death within seconds. And so how do you avoid those sorts of situations? And that's where the particles come in, is if you just deliver the peptide, they can interact with cells in the blood that can lead to that anaphylactic reaction. But if we encapsulate them within particles, these particles avoid the cells in the blood typically, and actually are then directly delivered to the antigen cells in the liver and the spleen. And so what's happening is these particles are able to bypass the cells in the blood and are actually able to get to the cells in the liver and the spleen, and actually able to present the antigen without inducing the co-stimulatory signals that would normally induce an immune response. And therefore we believe we get a tolerogenic response, which is shown here. So the particles taken up by APCs, they present the antigen, they secrete IL-10 and TGF-beta, but they don't present the negative co-stimulatory factor, or sorry, the positive co-stimulatory factors. Essentially, they don't activate T effector cells, they lead to energy and deletion, and actually can induce the development of regulatory T cells. So that's how we believe it's happening. And I'll just end with this last thought, where we're actually saying, well, can we take the same technology that we applied with the P31 being a single antigen and tolerizing to that, and think about, could we do this with allogeneic cells and take all the antigens that are unique to the allogeneic cells and put those onto the particles, and how effectively could that work? And so this is the illustration here where we take the spleen from a bulb seed donor, we isolate the cells and cell lysates, we put those cell lysates into particles, and then we do a tail vein injection into a black 6 mouse, at which point we follow that up with a transplantation of islets. You can see that essentially the strategy here. And when we had done this directly, we were only slightly discouraged to see that about 20% of the animals actually had a long-term engraftment of the islets. So while not successful, I will say at least I took away as hope and potential for this. And we did a series of studies where we looked at, and we noticed that one of the major challenges was that our particles aren't really very good at attenuating a direct antigen presentation, which means that the passenger leukocytes that are coming along with the islets are able to come out and essentially incite an inflammatory response. So what we had done is, instead of redesigning our particles altogether, we essentially added a short course, a 3-day rapamycin treatment around the time of the transplanting of the islets. The idea was try to use that rapamycin treatment to essentially deactivate or kill off those passenger leukocytes that could be initiating a response, and then using the particles to actually try to tolerize the indirect antigen presentation that comes from the shed antigens being presented. And you can see by applying this approach, we're actually able to get up to a 60% engraftment of the islets for a relatively long term, whereas essentially the other control studies that were done here really didn't have a significant engraftment. So with that, I believe my time is up. I will just highlight essentially the biomaterials that we've been using to sort of create an environment for transplanting cells, or maybe promoting their differentiation and their engraftment. At the same time, also thinking about the immunomodulation of modulating the responses both locally for a systemic response, but also maybe having more broad-reaching systemic uses in terms of nanoparticles to modulate what's happening there. So with that, I'd be happy to take any questions, and again, greatly appreciate the opportunity to present to you today.
It was fantastic presentations. I don't want to damper it either here, but my question was, the immune system is so different in a mouse, and you used a couple different strains there. So how do you get over the barrier to translate this into a human population or something that has more variation?
So there's obviously a very complex answer to that. And so I will say that there are some people who are arguing for that rather than like a pluriform stem cells or iPS cells, that you have these banks and you do your somewhere between 20 and 80 cell lines to cover most of the US population. And so you essentially have cells that are HLA matched and cover most of the people that way. Then you have minor antigens that you still have to deal with. And so that's one aspect of this is simply is, well, are there things that you can do to make it such that this immune response isn't so robust, but might be something that's more controllable? And that's sort of one option. I can say there are certain things like Fasl ligand are sorts of conserved between species. And so Fasl, my collaborator, is actually doing parallel sets of studies in non-human primates with that and seeing some things. But you're right, there are differences in there. So while it may not work exactly as you would see in the mouse system, I think that it becomes a starting point for beginning to think about how you might do those things. And at the same time, the last part I can add is this approach with using the cells as the carriers for antigens. There actually was a clinical trial that was done in Germany through Steve Miller, my collaborator, and German clinician on multiple sclerosis, where they actually had used peripheral blood cells as the carriers for antigens, and they're actually able to see tolerogenic responses in them. So while I don't think everything is going to be perfectly smooth, I think there's a reason to think that these technologies can translate. And you're right, how do we go from one to the next? Well, that's why I think I'm hoping to have a conversation with people here about what's the right way to approach these large animal studies.
I just have a quick question. We know, as you said, that the biggest problem with our transplantation is that the islets are dying up to 60% after transplantation in humans. And we see pretty much the same number in animals as well, if no methods are applied to protect them. So in some graphs that you showed, we saw 100% islet survival. That's what you showed. Do you?
I think there probably was graft survival. So 100%
graft survival. Okay. So what do you actually mean by that? That's one question. Do you really mean that 100% of the islets are alive? And the second question is, how did you measure that?
So what that is intended to mean is that 100% of the animals were not diabetic. I do not in any way think that all of the islets survived. There certainly is some that are dying out.
Do you know how many in comparison? Just curious to compare to non-protected.
I would say that we are, with some of the islet work that we've done in the mouse, I think we're probably up to 50 or 60. I'm used to, like, you transplant things into the liver, that you can get 60 to 80% death within the first couple of days. I don't think we see that because we're not in the vasculature. We don't have that blood-mediated inflammatory response and everything.
But they have hypoxic too.
Right, but they are hypoxic, but and some die off. But they do. I think that's why the apposition to the host tissue is so key, because they can get fed that way. Again, people are transplanting islets encapsulated with an alginate, and they can never revascularize, and yet they can still get some survival and function of those. So there's a window of time that that things can survive, and you try to minimize that window as much as possible.
So how did you measure that?
The measure.
Survival. Oh, so you didn't prove that the viability of the
We have not taken them back out and counted the islets. It's a very challenging study to do.
All right, let's thank the speaker.